Okay, so we are going to learning objective. After this chapter, you must be able to read. Yes, so fast and slow excitatory. Excitatory means inside become more positive. Inhibitory means inside become more negative due to local this potential. And fast means channels are fast nature, how much time it is taking. And explain how potential interact to generate action potential. All the local potential not causing the action potential, right? Define and give example of direct inhibition, indirect inhibition, presynaptic inhibition and postsynaptic inhibition. This is you will learn in neurosciences, but it is better to learn this time. So describe neuromuscular junction, explain how action potential in the motor neuron at the junction lead to the contraction of the skeletal muscle. Define denervation hypersensitivity, different synaptic transmitters in terms of signaling function and their role in the disease. Most of the diseases, neuromuscular disease we say, because of this neurotransmitter problems. Describe electrical and chemical transmission, how they differ, right? And how the specialization of synapse contribute to the synaptic transmission. So what is synaptic transmission? Neurons communicate with each other through synapses, through the junctions between them. So neuron, two junctions are called synapses, right? So impulses are transmitted over chemical or electrical synapses. Chemical synapse means they are the normal, most of the synapses are chemical synapses. Means there is a chemical communication between the two neurons. They are talking with the chemical. Another is electrical synapses. They are linking one neuron to another neuron, like muscle, gland. Okay. Secretory vesicles. Do you know what is secretory vesicles? At the axon terminal, they are synthesized into the cell body or locally there, and these amino acid, proteins, a specific type of chemicals, they are packaged, they are synthesized and packaged into the vesicle. When the action potential comes at the axon terminal, right? Action potential is by which channel? Till now you studied. First, which channel will open? At the action potential. Voltage gated sodium channel. They will cause depolarization. By the time they will close, another set of channel will open, that is called voltage gated potassium channel. They will cause hyperpolarization. So this is the pattern, right? So you can record it anywhere. Yes? Oh. Okay. So what happens? They are neurotransmitter and rest in the cytoplasm of axon terminal inside it. When this voltage gated sodium, calcium channel open, along with that at the axon terminal they contain calcium channel, voltage gated calcium channel. So voltage change happening with the sodium channel, then what is happening? Calcium is also entering into the cell, and calcium having sites there, calcium binding proteins. When they bind, they will activate it and they will cause docking. This is called docking, right? They will bring this vesicle to the nearby to the membrane. So when they attach to the membrane, they will release it by exocytosis. So it release into the exocytosis of these secretory vesicles, and their secretory vesicle, they contain neurotransmitter, right? They release from the presynaptic axon terminal, right, by the process of exocytosis. So intercellular communication, how two cells communicate at the synapse, right? I think figure there. Okay. Here two neurons coming, right? One from here impulse coming, right? They are dendrites. Then if they are reaching the synaptic, axon terminal, we discussed, right? From axon terminal there is generation of action potential. Action potential will travel till the end here. They are ending. This is called axon terminal, right? Then another cell here, they will make a connection. This is called synapse. So from here the message coming in the form of action potential, right? Action potential. This action potential, right? What is event here? This is called presynaptic cell. Presynaptic cell. This is called postsynaptic cell. So what is the event here? Action potential. Action potential is causing voltage change. Voltage change here, along with the sodium potassium, there is another special channel which is called voltage gated calcium channel. That is important, right? All the presynaptic terminals, they have which channel? Voltage gated calcium channel. So voltage gated calcium channel, right? They will cause entry of calcium, and this calcium will bind with the what I told, proteins, right? Proteins which will cause vesicles, a small vesicle to go into the membrane area, and they will release by the which process? What is the process of this release called? Exocytosis, right? Exocytosis. Is it clear, Latif? Did you get it? Look, I am explaining here. So this is the one cell. So what happens from here? Different local potential will travel, right? And so this is called local potential. Why? Because they are not voltage gated channel. They are the some other mechanical gated, etc. They will call local channel. And from here, voltage gated channels will start. Here they are located, right? Which channel? Sodium, potassium, etc. So if they are reaching the threshold, they will cause action potential. No threshold, no action potential. They will die, right? And in the last here, suppose I am making here blind, and another terminal, another cell here, right? This is called presynaptic terminal. This one is postsynaptic. Right? And this is called synapse. And gap between this is called synaptic cleft, right? Synaptic cleft. Here it contains sodium channel, voltage gated, right? Voltage gated sodium channel, voltage gated potassium channel, and especially this area here, there is, along with these two, they are having added thing is calcium, voltage gated calcium channel. So when this, and before the action potential happens something, they contain a lot of this vesicle containing neurotransmitters. So they are not released until and unless there is a special mechanism, that is the action potential. So when action potential comes, they will cause entry of calcium. Inside the cytoplasm here, calcium will increase, right? So calcium will bind with some special proteins there. They will bind with them and they will cause this vesicle to move, right? They will pushing the vesicle towards this one, right? So what will happen when they come in? They will release their content by exocytosis. So this will result into the vesicle movement and release of what causes this action potential in presynaptic terminal, right? Presynaptic terminal action potential, then they will cause entry of the calcium, then calcium is binding and causing a release of the vesicles. Release of the vesicle means release of the chemical, right? And this chemical may be linked with the calcium channel, sodium channel, potassium channel, right? Suppose it is linked with the sodium channel here. This is released here, right? It is released. Then they have receptor on the postsynaptic terminal here, right? They will bind on the postsynaptic terminal, right? They will bind with the postsynaptic terminal, and these receptors, they are linked with the channel. So binding of chemical, where it is binding? Neurotransmitter, where it is binding? Postsynaptic terminal, right? Postsynaptic terminal. What they will cause? They may be like sodium channel opening, then they will cause depolarization, right? Depolarization means excitation or inhibition? Excitation. So it is called excitatory postsynaptic potential, right? If this transmitter is linked with the chloride channel, what will happen? Chloride is more outside the cell, it will go inside. Chloride if entering, what will happen? Cell become more negative. It is called inhibitory postsynaptic potential. If it is linked with the potassium channel, extra potassium channel is opening, right? It will cause exit of the potassium. It is inhibitory, right? So it depends on what type of they have machinery, inhibitory or excitatory, right? And this is the local or action potential. Is it graded potential or action potential? Graded, right? Because it depends on, it is going in both sides, and how many channels open, that is also variable according to the chemical, right? So what will happen? They don't have here voltage gated channel. They will go with the decreasement till here, right? This is the example in this figure. So if here it is reaching threshold, they will cause action potential, or it will die at least, right? So this is the area of the junction, is the area where anything can happen. It will die out, it will be getting more inhibited, or it is getting more excited, right? Like you have to change the bus, right? You have to go somewhere. So you have to catch the bus, go to the junction, then catch another bus, right? So when there is junction, you change the mode. Oh, I am going to just out, that I can go to the Mesopotamia. You can catch another bus. So at junction is a site of modulation. So you know the term modulation is change, right? They can change it, right? So something, our mood, everything is by this chemical transmission, right? So what is happening here? Local change, Okay, so here you got what is synaptic cleft, right? The gap between the two sides. Postsynaptic membrane, this one. Presynaptic membrane, this one. Right? Receptor means where the chemical binding, and this receptor is linked with the channel. They may be linked with the channel. What type of channel? That depends on type of receptor, right? For the same chemical, in some cell it is inhibitory, another cell it is excitatory. Like acetylcholine is a very common neurotransmitter. Another common neurotransmitter is sympathetic. Which chemical is called sympathetic? Epinephrine and norepinephrine. So epinephrine, norepinephrine, they have receptor, excitatory or inhibitory. That is quite variable. Like when you are having an emergency, your heart become more excited, right? Increase, heart rate increase, right? What will happen to digestion? Suppressed. So same chemical there. Here it is only excitation. Why? It is linked with the which channel? It is causing excitation, increase, same thing, same chemical when binding in your digestive muscles, right? Muscle of the GI tract, it will be slow. So how it is getting slow? Potassium channel or chloride channel, right? Or closing of the sodium channel. Sodium is going on, it will close it, right? Two-way, right? Closing of the sodium channel, it is inhibitory, right? It is more permeable if you are closing it. What will happen? It will become more negative, difficult to excite, right? So these are the chemical transmitters. So these are the points we have to know. They told, okay, so ion channels, you got it. Potassium channels are responsible for depolarization. No. This is wrong, right? In action potential. Which one is responsible? Yes, sodium channel. That's we have to think. This is showing gap channels in opposing membrane. Gap junction channel. This is, our heart muscles, right? Our heart muscles, these cells are linked with each other, right? What is the term called? This gap junction, interdigitation something, right? So these cells are linked with each other with a channel, right? So what happens? They are like electrically coupled with each other. Something like this room is connected with an interior gate to another room. So they are like single room, right? Attached type. So if something change happens, it will be reflected there. So when you changing permeability in this one is getting depolarized. What will happen? They are easily getting transmitted to other cells, right? So they are electrically coupled. That is called electrical synapses. Electrical synapses means they are almost behaving like a single cell. Electrical synapse means neurons communicate by electrical and chemical synapse. So electrical means you don't have to excite, you don't have to release the chemical. One cell is when they are connected with another cell. Suppose this is connected with this one, another cell here. What will happen? They don't have to release the chemical. When voltage gated channel coming, it will spread here. Right? This is called electrical synapse. Yeah, directly they are linked with each other with some bridge. That is called connection. These are found in like heart cells, some special part in the brain.
So saying the action potential, the electrical
Yeah. Yeah, no, it's cytoplasmic correction directly. there is cytoplasm is connected with each other. So this when actually this action potential is causing change here, ionic change, right? So this ionic changes is going directly, not through the membrane, directly through the cytoplasm, into the electrical junctions, electrical synapses. So electrical synapses is electrical communication via gap junction. What is gap junction? Like a bridge between the two cells. Ion flow from one neuron to other directly, right? Cytoplasmic exchange without release of neurotransmitter. You got it? Without release of neurotransmitter. So gap junctions are found in the embryonic nervous system, but rarely occur in adult mammalian neurons. This is rare, right? I told where it is common is this type of electrical junctions, heart muscles, right? In the all cells in the ventricle, they are acting like a together, right? That is called single unit muscle. They act like a one cell, single unit. So a smooth muscle is two types: single muscle unit and multiple unit. Cardiac and single unit smooth muscle cells have these electrical synapses, electrical junctions, right? So you can see here, this is the presynaptic cell, postsynaptic cell. They are directly linked with each other. No need of chemical communication. Here, this is releasing the presynaptic mitochondria. They need to synthesize this one. Synaptic vesicle, right? So this is the what type of junction? This is called synaptic transmission, synaptic junction. This is the chemical synapse. This is the electrical synapse. Direct electrical activity changing, right? Ionic influx directly. So presynaptic, postsynaptic, right? Active zone means where it is like a warp, you know, where the boats coming, right? This is called active zone, means where they exactly fit the vesicles, they will bind, because they need a special rope to tie and cause EG, right, passenger and other things. So similar here, vesicle will bind to the active site and they will be released. What causes this pushing of the vesicle? Huh? What? Asim? What causes this vesicle to come nearby to here? Action potential, right? Action potential, entry of the calcium. Right? You telling directly calcium, right? Yeah, good. So postsynaptic density here, postsynaptic, they contain receptor for these chemicals. So this is showing type of the synapses. Synapse possible there is, like this is the cell. Exodendritic means this is the axon, and telling something. Okay, so now chemical synapses. This is focusing on the one, and what I told you. What is number one? Here indicating. This is indicating formation of vesicles with a neurotransmitter. Neurotransmitter is locally, or they are synthesized from the cell bodies. It depends on type of what type of chemical there. If it is very complex, large, they need cell bodies, right? And they are transported from there. And then it is remaining here, waiting, right? These vesicles are waiting for the action potential. Number two indicating action potential. When action potential comes, what will happen? series of the depolarization, repolarization, hyperpolarization, right? So they have, what is special like the axon terminal? What is here focusing? Which type of channel this one? Voltage gated calcium channel. So when they are getting voltage change, depolarization, they will open. They will cause entry of the calcium. Calcium bind with the vesicle proteins, right? Synaptic vesicle trafficking protein, and they will cause this exocytosis, right? They release, and here look, they are binding. These chemicals are binding with their receptor. This receptor is part of the ionic, right, bridge. They are part of the ion channels. So when they bind, or they look here, adenyl G protein coupled receptor here. This receptor is also bind with the channel. So anyhow, this channel will open. They may cause depolarization, hyperpolarization, right? Anything. And this is the synaptic transmission. So chemical synapse contains presynaptic terminal, synaptic cleft. Cleft means gap between the two, right? Postsynaptic terminal, right? So these are the sequence of events we have already discussed, right? Look. So synaptic transmission between the neuron. Look here, this green color, they are showing, what they are showing? They are the presynaptic cell or postsynaptic cell. These are presynaptic, and this is the postsynaptic. Okay. Suppose one is causing, here only change in the 1 volt, right? So if all will be fired together, so 3, 3, 6 to 8, right? Suppose each one is 2 volt. So if they are firing separately, they will not cause action potential here, right? Suppose you have 15 millivolt your threshold. So what will happen if all are firing together? They will cause 15 millivolt change, right? So this is called summation. Initial segment, myelin sheath, axon, vesicle transmitter. So if neurons are of two types, myelinated and unmyelinated. If they are myelinated, wrapped with the fat here, fat layer, that is called myelination. So what will happen? Channels are located only at the gap junction, nodes. This is called node. At the regular interval, there is gap between the myelination. So they will jump, ionic influx here, then they will jump, right? So it becomes very fast. Action potential, conductance of action potential is very fast. If it is unmyelinated, a small diameter, it is very slow. It varies from 0.1 meter per second, like in unmyelinated C fiber. According to this neuron axon diameter, neurons are classified. Very large diameter, very fast ion conductance. Large diameter and the myelination. A small diameter, unmyelinated, very slow. It will go opening individual ion channels, and it is taking more time. So series of changes. Normally what happens? Outside is positive, right? Inside is negative, minus 70. When this is spreading, what will happen? This is causing sodium entry. So inside become positive, outside become negative. Right? Reversal of polarity is called action potential. So it will spread here. This is showing example of the acetylcholine release that is happening in case of muscle. This is the neuromuscular junction, right? What we describe same thing here. Action potential coming from the motor neuron, right? Number 2 event, calcium entry by voltage gated channel. Then number 3, there is release of the at the neuromuscular, right? Voluntary muscle having at the neuromuscular junction, what chemical they are releasing? All are having same mechanism. Acetylcholine is a transmitter, right? The receptor they bind, it is called cholinergic receptor. They are called cholinergic receptor. They are binding here and they are causing entry of the calcium. When they are reaching the threshold, look here. On the side, here it is the chemical, this is the chemical gated channel, right? Acetylcholine channel is chemical gated channel. And they are having on their side, what type of channel here? Voltage gated sodium channel. So when they are reaching threshold, they will open. No threshold, they will not open. When this opening threshold, muscle fiber action potential they will cause. Right? They will cause. So local current between depolarized end plate and adjacent muscle plasma membrane. When they are reaching voltage gated sodium channel open, right? Muscle fiber action potential initiation. Right? And later it is very quick process. Acetylcholine release, they will getting degraded by the another a lot of chemical here. This is called acetylcholinesterase. That will degrade the acetylcholine into the acetyl and choline. Right? So choline is taken back to synthesize the another vesicle, another neurotransmitter, and other is, so it is quick. When it is released, either it is binding, rest of the thing is getting degraded. For your knowledge, this binding protein, I told calcium is binding with a special protein, right? This protein name is SNARE protein, like NARE, soluble SNARE, sorry, S here missing, SNARE protein, soluble N-ethylamide sensitive factor attachment protein receptor. It is difficult, right? It is short called SNARE protein. You can see here. So when the calcium bind, and this is the vesicle, right? Synaptic vesicle. It is fixed, right? It is fixed with the which protein? SNARE complex. It is tied. So when I think another here. So synaptotagmin 1 calcium sensor, when they have calcium there, they will be binding here. Then what will happen? They will pull it. They will cause conformational change and they will cause binding of this synaptic vesicle here. Exocytosis. In short term, it is called causing exocytosis. Right? Okay. This is more neuroscience, but it is interesting. Receptor type. Receptors can be divided into this is a molecular receptor. This can be two types, inotropic and metabotropic. Inotropic means linked with the ion, what we discussed, right? Linked with the ion, like acetylcholine, this sodium gated choline channel, choline, right? So it is called inotropic. A synaptic response generally fast. It involves direct opening of a channel by transmitter. Okay. Metabotropic receptor is this metabotropic means receptor is binding and that is causing metabolic changes into the cell, right? Some that is called second messenger formation, and this second messenger will cause opening of the channel or doing something, right? That is called metabotropic channel receptor. They involve activation of second messenger, and then second messenger will do the activity. So ligand gated ion channels are directly, look here, they will bind with the channel, then they will cause opening of the channel, right? Normally they become narrow, they will not cause ions to pass. When they binding with the neurotransmitter, they will cause opening, or some may opposite event happen. They are open, when bind they will close it. Metabotropic, you can see here, metabotropic is they are binding with the receptor, G protein, right, chemical. So this chemical indirectly they will bind here and coming the opening or closing of the channel. So excitatory neurotransmitter, we say, what is happening with the excitatory? They will cause inhibition, depolarization or hyperpolarization. Excitatory depolarization. So depolarization is by either by the sodium opening of sodium channel, opening of the calcium channel. Indirectly means potassium channel become closed. So it will cause the same depolarization, right? Calcium channel open by other means. G protein here, metabotropic receptor, they will causing binding of the receptor and formation of G protein activated. They will having different subunits. So one of the subunit will cause opening of the ion channel, right? So it will take more time. Here it is showing nicotinic receptor for the acetylcholine. Acetylcholine having two type of receptor, nicotinic and muscarinic. They are named according to other chemical can excite this. So some acetylcholine channel they are can bind with the nicotine. This is the part of the tobacco, right? So they will bind and they will cause opening, excitation of this receptor. So it is called nicotinic. But some are not responding to that. Those who are not responding to nicotine, they respond with another mushroom chemical that is called muscarine. So they are called muscarinic receptor. I think this is the repetition. So a lot of information in slides. So go through that. Excitatory means it is causing firing of action potential. More likely, right? Sodium channel, opening of sodium channel, calcium channel, or closing of the potassium channel, closing of the chloride channel. They cause excitatory postsynaptic potential, right? Important receptor that produce this excitatory postsynaptic potential is nicotinic acetylcholine receptor, right? Endogenous ligand is acetylcholine and include neuromuscular junction, right? Neuromuscular junction or neuron junction, NN. Non-NMDA receptor like endozygon. Another type is for the excitatory postsynaptic potential is non-NMDA receptor. That is NMDA belongs to the, it indicates N-methyl D-aspartic acid. That receptor also binding with this chemical, right? So non-NMDA endogenous ligands are glutamate and aspartate. So glutamate and aspartate is excitatory amino acid transmitter. So when they bind, what will happen? They will causing depolarization, right? Sodium conductance increase when they bind. What they telling? What you telling? Break? Okay, break. Take a break. Inhibitory postsynaptic potential is it is causing inhibition. So transmitters hyperpolarize the cell by opening up the what type of channel? Chloride channel or possibly potassium channel, right? Efflux of the potassium or entry of the negative chloride channel. Transmitters, inhibitory transmitters in our body mostly include GABA or glycine. They are very common. So they are acting through, look here, they bind, this is the chemical binding site. When they bind, they will cause opening of the either chloride channel directly or through the indirectly opening of the potassium channel, calcium channel, closing. If sodium channel there, they will close the sodium channel, right? Indirect means they are acting through the G protein or some other second messenger transmitters. Look, so this number A, resting neuron, minus 64, 5 millivolt. If it is excitatory means it is causing decrease in negativity, right? And if it is reaching threshold, they will cause action potential or some additional they require, then two maybe synapse firing together, right? That is called a spatial summation. Two at the two different places, if they are firing, what will happen? They will getting added. That is called a spatial summation. Summation is of two types. One is a spatial, different spaces, different places firing together. Like one is here, another is here. If it is causing here, how much? 65 to minus 45 means 20 millivolt, right? If your threshold is suppose 40, they need to reach minus 40. Is this causing action potential? No, right? They need one additional. So if another neuron here, they are firing together, right? They will cause more than 25, and so this type of summation is called a spatial summation. Another is if this is firing continuously, how much time it is causing to cause one action potential? How much time it needs to cause one action potential? 2 to 5. So if within 2 to 5 millisecond another action potential means sequential, to very fast, then entry of the sodium before it is going back, entry of the, it will cause another more channels open, right? This is not by the voltage gated channel, so it is by the chemical gated channel, right? So if it is reaching, this is called temporal summation, time pattern, sequential, right? So two type of summation. If chloride channel is opening, what will happen? It is causing hyperpolarization, right? This is called inhibitory postsynaptic potential. This is showing neuromuscular junction. Neuromuscular junction always for the skeletal muscle, what is the transmitter? Chemical acetylcholine. What is the receptor here? Neuromuscular, neurotransmitter, gated channel, right? They are the called motor end plate. You can see here motor end plate. So this is called motor end plate potential. And not action potential. This is not action potential. Action potential starts from here. Why? Because you need what type of channel? For action potential, voltage channel. So look here. This is showing miniature end plate potential. You know, I think, what is miniature end plate potential, right? What is that? Even you are not firing action potential. Some very few, one vesicle, two vesicle, they are released, right? They are causing here local potential. That is not reaching the threshold. That is called, what is named for them? Miniature end plate potential. Look here, this is only 0.5. But you need threshold how much? Like 5 millivolt, right? Then it is causing action potential. So action potential evoke 200 quanta. Quanta means 200 vesicle release, right? One is causing 0.5, means more than, it is very strong, right? So what is here? Central neurons integrate a variety of synaptic inputs through the temporal and spatial summation. We just described what is temporal and spatial. Spatial means different synapses, like this one. You can see here only one synapse firing repeatedly. That is called temporal summation. Many synapses fire at a time, different spaces. That is called spatial summation. So look here, one firing, another firing. Here, two at a time, right? So when there is synaptic current there, one is not sufficient to cause firing, but before it will die out, before it is coming to normal, again another firing here, another local potential. So that will added up and then it is causing action potential, right? So here if one is firing, no action potential. Where another is firing, there will be action potential. What here showing? Number A. A is single action potential. Single binding. It will not cause action potential. When it is reaching threshold, it will cause the action potential. Right? So amount of calcium influx correlates with probability of neurotransmitter release. You know where the calcium is going? What is the role of calcium? Where it is calcium? Calcium. Where it is going? Where the voltage gated calcium are located? Presynaptic or postsynaptic? Yes, presynaptic, right? So amount of calcium influx, how much calcium going there? That is correlated with how much neurotransmitter release, right? More calcium going, more release. Less calcium going, less release, right? Large influx of calcium during action potential causes synchronous, means at a time, release of 150 quanta, 150 vesicle to ensure that end plate potential generate an action potential. That is the safety factor. You need 100, but they are giving more, right? So that your muscle, when you are trying, your muscle should contract. A conotoxin from cone snail venom often block currents in the presynaptic terminal to decrease neurotransmitter release without inhibiting the action potential. Right? Conotoxin, you can see these are the snails, right? These snails release venoms which will paralyze their prey. Right? So their mode of action is these venoms are acting, blocking the decrease of neurotransmitter release. So which is directly responsible for the release of neurotransmitter? Sodium, potassium, chloride, calcium, both sodium and calcium. Which one? Yes, calcium is the. So conotoxin block voltage gated calcium channel to inhibit neurotransmitter release. Neostigmine, right? Here stimulate the nerve, right? So control, when you are giving a stimulus of high millivolt, it is causing the action potential, excitatory end plate potential, right? But when you added the curare is the chemical, right? Then it is blocked here, right? No action potential. Another thing, control action potential. If you are giving neostigmine, it is causing, right, more large action potential here. So this modulation of end plate potential. Curare blocks neuromuscular junction nicotinic channel, right? They will block it. They are going binding the site, so no available for the. It is not available for the acetylcholine, right? So what will happen? No action potential into the voltage gated channel. Neostigmine like pyridostigmine, they block acetylcholinesterase. We discussed that, and the synaptic collapse when chemical coming, right? Either they binding or they are getting destroyed. Acetylcholine is getting destroyed very faster. If they are not binding, rest of them they will getting destroyed by which enzyme? Acetylcholinesterase, right? So if this acetylcholinesterase is blocked, what will happen? They are not getting destroyed. Acetylcholine release, they can act for longer time. When they act for longer time, what will happen? Action potential will be strong, right? Action potential will be strong. This is showing the summation. If one is causing here, one is firing, A is firing, and it is firing it up with a little bit longer gap. So one firing, it is causing depolarization, local depolarization, right? Again it will come to normal, die out. When you are giving another firing, again it is showing same, no action potential, right? When you are firing only A in sequence, then what will happen? It becomes stronger, right? Then B also, so B having how much here? This much it is causing. A and B together, this one. But when you are firing A and B repeatedly and both together, what will happen? They will cause action potential. So this is called a special when two together and repeatedly, this is called time pattern is called temporal summation. Okay, this is showing presynaptic inhibition. This is here is the cell, it is releasing the chemical, right? And this chemical will bind here and it is causing response, presynaptic, postsynaptic, right? When this B is action potential coming, they will cause release of the chemical, they will bind here, that is causing depolarization, right? But when the B is firing at the same time, when A is also firing, what will happen? And this is so for this A is presynaptic and B is postsynaptic, right? And for this C, B is presynaptic or postsynaptic? Presynaptic, right? So when A and B firing together, A is causing, suppose it is releasing the inhibitory chemical which will bind here. So what will happen? It will try to inhibit. When it is inhibited, the release of the chemical will be less, voltage change is less. So it is called presynaptic inhibition. It is called presynaptic inhibition. So what will happen to the postsynaptic? It is also getting inhibited, more inhibition, right? A drug might, when you are using different drugs, their mechanism is, we just saw the snail conotoxin. It is causing blocking the calcium release, right? Another way, another drug we saw pyridostigmine. It is blocking the acetylcholinesterase. So action potential will be more. It is causing more excitation. If you are exciting for longer time, what will happen? It is getting inactivated, right? Inactivation get, so it will getting paralyzed. A spastic paralysis or flaccid paralysis. Both are same. So drug you are using for to change the synaptic activity may be different type of action. Like number one, a drug might increase leakage of neurotransmitter from vesicle to the cytoplasm, exposing it to enzyme breakdown, right? Then if it is getting broken, what will happen? There is no release, no action. Increase transmitter release into the cleft. If increase transmitter means more action. Block transmitter release like conotoxin. Inhibit transmitter synthesis. How much synthesize? Synthesis itself block. Then what will happen? Release is less. Voltage is coming but no chemical. Inhibit transmitter synthesis, block transmitter reuptake. For resynthesis you need reuptake, so that may be possible. Block cleft or intracellular enzyme that metabolite transmitter, like blocking the pyridostigmine. They are blocking the acetylcholinesterase, so they can act for longer time. That is used in the resistant diseases like myasthenia gravis. Bind to receptor on postsynaptic membrane to block, they are binding and blocking it. That is called antagonist, or they will enhance it, mimic the transmitter action, that is called agonist. Or finally inhibit or stimulate second messenger activity, that is the metabotropic receptor. So these are, we are giving the trying to modulate the synaptic activity. If someone is very excitable person, what type of drug is given? Like some psychological diseases, they are more excitable, right? We saw many people more excitable, firing. So what type of drug you need? Inhibitory or excitatory? Very good, inhibitory, a GABA type of drug, right? Inhibiting it, slow down. And some people so sluggish, coma, then what type of drug you need? Excitatory type of drug you need. So these are the points. So postsynaptic factor, like immediate past history of electrical state of postsynaptic membrane. Excitatory, if excitatory is going, another will come, it will enhance, like temporal or spatial summation. Effect of other transmitters or neuromodulators acting on postsynaptic neuron, up or down regulation and desensitization of receptor. Certain drugs and diseases. General factors, area of synaptic contact, enzymatic destruction of neurotransmitter, right? Geometry of diffusion path. If it is going longer time, longer duration, longer gap, what will happen? It will slow down, right? It is getting destroyed before it is reaching to the site, area of action, right? Botulinum toxin. You will get a lot of questions regarding this action of the pyridostigmine, conotoxin, botulinum drugs, right? These are the points you have to remember. Like botulinum, Clostridium botulinum, a gram-positive bacteria, produces a potent toxin that is called botulinum. What happens? They prevent release of neurotransmitter in the CNS and at the neuromuscular junction. So if neurotransmitter is not released, what will happen? Action will less, right? So it is caused by the contaminated food, colonization of the GI tract in the infant or wound infection. So what will happen? They will, botulinum toxins, the A and E cleave synaptosome associated protein, which is caused the release of the neurotransmitter. We discussed that calcium is going binding with the protein. So what will happen? It will presynaptic membrane protein needed for fusion of synaptic fascicle, they will block it, right? So what will happen? They will block transmitter release. So it is causing paralytic type of disease. Botulinum A and E. Botulinum B cleaves synaptobrevin, a vesicle associated membrane protein. So this is causing flaccid paralysis, means in resting condition muscle is weak, muscle is relaxed and paralysis, no activity. You are trying to contract but it will not respond, right? Neuron is firing, your neuron is firing but no chemical message in between. So it will cause botulinum toxin, ptosis, drooping of the eyelid, double vision because this muscle is not working in the eye. You cannot arrange. Dysarthria means speech, slurred speech. Dysphonia, difficulty in speaking, difficulty in eating, dysphagia, swallowing. So local, but this botulinism, so these are the poison disease, right? But clinical used, this is used clinical, like venoms, snake venoms are also used. So local injection of a small dosage of botulinum, Botox injection is given, right, for the treatment of the muscle hyperactivity, right? And lower esophageal sphincter relieves achalasia when they are getting excessive contraction of the esophageal muscle. What will happen? They are not getting relaxed, so that food can go, right? So they will cause burning sensation. right? So muscle is relieved by this one, and to remove the wrinkles they are used into the muscles. Myasthenia gravis, this is a very common disease. I think you might have studied before. This is by the antibodies. This is an autoimmune disease. They are forming the antibody, destroying the muscle nicotinic receptor. We discussed that neuromuscular junction, nicotinic receptor there, which is causing muscle contraction. So if the receptor number is decreasing, what will happen? You are trying to contract the muscle, they will not work. Especially in the myasthenia gravis, it's become more worse in the evening. In the morning, neurotransmitter release is more, so that will act. As the days goes, right, person become more tired, right? So extraocular muscle primarily affected, generalized skeletal muscle weakness, right? So appearance of a sparse, shallow, abnormally wide or absent synaptic cleft in the motor end plate in this disease. So what is the treatment? Administration of the neostigmine or pyridostigmine. What they are doing? They will stop the breakdown of the neurotransmitter, acetylcholine. So they can act for a longer time. Person will feel very relieved, right? Like normal when they are injected with the neostigmine and pyridostigmine. Or immune suppressive drugs is given because it is a autoimmune disease. Lambert-Eaton syndrome. This is 1 in 100,000 population into the US. It is common. So it is a muscle weakness. Again, similar to the myasthenia gravis, but this autoimmune is attacking the voltage-gated calcium channel in the nerve ending. So what will happen? Less voltage-gated calcium channel, less release of the neurotransmitter. So neurotransmitter there, but it is not release, right? So decreased calcium influx needed for the calcium acetylcholine release. So proximal muscle of the lower extremities are primarily affected, right? And it is a waddling gait, difficulty raising the arms there. So for that, to differentiate between the Lambert-Eaton syndrome and myasthenia gravis, both are muscular weakness, right? So administration of iduronium, a short-acting acetylcholine inhibitor, rapidly improve the myasthenia gravis, but it is not improving the Lambert-Eaton syndrome because chemical, right, is not released here. So no use of acetylcholinesterase blocker. Okay, that test is called tensilon test. So what is therapeutics for the Lambert-Eaton syndrome? They are in patients without cancer, immunotherapy is inhibited. Prednisone administration, plasmapheresis, right? So they are given there. So Lambert-Eaton syndrome, autoimmune, right? Suppressive drugs are given. Any question? Any question? Look here. How many synapses you can see here? Right? So this is the situation. A lot of figures here. You can read yourself. Right? Okay. Synaptic transmission, effect of acidosis or alkalosis. This is a very good thing. We have many times we are getting acidosis or alkalosis, means change in the pH of plasma. Generally, normal plasma pH is 7.35 to 7.45. If it is less than that, it is called acidosis. If it is more than 7.45, alkalosis. So normally, alkalosis greatly increases the neuronal excitability. H plus ion, right? It is H plus ion. When alkalisis means less H plus ion. H plus ion is inhibitory to the release of neurotransmitter or excitability, voltage gated channels, right? When H plus ion there, sodium channels are less sensitive, right? So, more H plus ion, less will be the sensitive. When H plus ion decreasing, they become more sensitive. More sensitive means excitability increasing. They are easily fired, right? What?
Acidosis.
No, alkalosis. Just opposite you telling.
Acid is a hydrogen ion, right?
Yes, hydrogen ion.
Alkalosis, that would be more basic.
Alkalosis is H plus ion. Sorry, H plus ion decreasing. OH is the alkalosis, right?
OH minus is increasing.
OH minus increasing. pH is opposite, na?
Yeah, there's basic and there's acidic.
Yes.
Acidic.
Basic means H plus ion is less.
Yeah.
So that same thing I am telling you. You are.
Because I thought you said that.
Yeah, H plus ion decrease. No. Listen. I think I am not confused. I am sure. More H plus ion, it is decreasing the excitability. Right? Is it clear? So alkalosis means less H plus ion. Neurons are more excitable. Is it okay now? Okay. So arterial blood pH 7.4 to 7.8. Right? When it become 8, it causes cerebral epileptic seizure. What is seizure? You know, right? Epilepsy. All are firing together. These neurons firing together, that is the epileptic seizure. So when it become 8, they will start firing. Acidosis depresses neuronal activity, like coma. Coma is due to acidosis, right? Like diabetic ketoacidosis. Person will be go in the coma. When it fall from 7.4 to 7, usually causes a comatose state. In very severe diabetic or uremic acidosis, coma almost always develops. Effect of hypoxia. Neuronal excitability highly dependent on adequate supply of oxygen. Right? So cessation of oxygen for only a few seconds, complete inexcitability of neurons. Right? Brain's blood flow is temporarily interrupted because within 3 to 7 seconds person become unconscious. They are so much sensitive. Right? So drugs we discussed like caffeine, theophylline, theobromine, that will increase the neuronal excitability and they reduce threshold for excitation of the neuron. Strychnine, that increase excitability of neuron by inhibiting the action of normally inhibiting neurotransmitter substances. Strychnine, they inhibit the inhibitory activity, right? Like glycine in the spinal cord. Glycine, another neurotransmitter, I told, inhibitory. GABA, gamma aberone, aminobutyric acid, right? GABA. Most anesthetics here we are using that will suppress your neuronal excitability, right? You will not sense like pain, etc. So they will increase the neural membrane threshold for excitation, decrease synaptic transmission there, right? Like local anesthetics, they are blocking the sodium channels. So no sodium channel, no action potential, no transmission, right, of the impulse. So if neurons here block, what will happen? It will not go. So tetrodotoxin, saxitoxin, local anesthetics, they block fast sodium channel, preventing action potential, right? They are asking. So you have to spend hours writing again and again. Ciguatoxin, batrachotoxin, right? They are toxin that block inactivation of phosphodie. Inactivation block means they will excite, they will remain into the depolarization state for longer time, but they are not causing the another action potential, right? They are paralyzing. Botulinism we discussed, right? They are target cholinergic neuron resulting in flaccid paralysis. Laterotoxin, black widow spider, they open presynaptic calcium channel. So excess acetylcholine release, they will cause firing, right? And then pesticide, therapeutic agents that will block the acetylcholine storage, right? Resulting in prolonged action of acetylcholine in cholinergic receptors, cholinergic synapses. So these are the points. You have these slides, right? So go through it and we will discuss questions tomorrow. So this is a very, you will get in USMLE at least 5-10 questions on this basis. So it has therapeutic use as well as pathological symptoms. Any questions so far?